This is the lecture for Chapter 8, Cellular Respiration. This is the overall equation for cellular respiration. And the main purpose of cellular respiration is to take glucose, a high energy carbohydrate, and use that energy to produce ATP. So that is the overall purpose of cellular respiration. The general equation is as follows. The reactants are glucose, that's 6-carbon carbohydrate, and oxygen. After a series of reactions, energy is produced in the form of ATP along with the waste products of carbon dioxide and water. The flow of energy describes how energy moves through an ecosystem, and it's important to understand where cellular respiration fits into the flow of energy. So the flow of energy follows the first law of thermodynamics, where energy can be changed from one form to another, but it cannot be created or destroyed. So flow of energy begins with the sun, light energy, that's the original form of the energy, and photosynthesis will convert that light energy into chemical energy, into the form of glucose. And then when the animals eat the plants, when they ingest that glucose, then the process of cellular respiration is going to convert that glucose into ATP, and then the animals can use that for mechanical energy. And throughout this process, converting energy from solar energy to chemical energy to mechanical energy, every time energy is converted from one form to another, you lose some as heat. And heat is disordered energy, and it is increasing the entropy of the universe, so which moves from organized forms to disorganized forms. On an organelle level, you should understand how photosynthesis and cellular respiration work. Same basic idea with the flow of energy. Light energy comes in, the chloroplasts perform photosynthesis, producing the organic molecules and oxygen as a waste product. Then those organic molecules are going to go through cellular respiration, of which the mitochondria is involved. And during cellular respiration, the mitochondria is going to take those organic molecules and break them down to release energy. So the end product is producing energy in the form of ATP. And the waste products are going to be carbon dioxide and water, which can be used as the reactants for photosynthesis. And throughout this process, every time you convert energy from one form to another, you're going to release some energy as heat. And remember, heat is disordered energy, increasing the entropy of the universe. It's important to understand the relationship between the reactions of photosynthesis and cellular respiration. And this is the equation for photosynthesis, which I discussed in a previous lecture. And remember, in photosynthesis, the reactants are carbon dioxide and water. And then the chloroplasts are going to use the light energy to fix that carbon dioxide into a high energy carbohydrate, glucose, and the process releases oxygen gas as a waste product. Now, when you compare the equation for cellular respiration, what you should notice is that the two equations are basically opposites of each other. So in photosynthesis, the products are glucose and oxygen gas. In respiration, the reactants are glucose and oxygen. And then in respiration, after you run through the series of reactions, the products are going to be carbon dioxide, water, and energy. And if you look at those products, carbon dioxide and water, those are actually the reactants for photosynthesis, carbon dioxide and water. So you should understand the relationship that photosynthesis and cellular respiration, in terms of the molecules that are being produced, are exactly opposite. So photosynthesis, carbon dioxide and water are used to produce glucose and oxygen. Respiration, the glucose and oxygen are going to be broken down to form carbon dioxide and water. So this is very cyclical. This is a very cyclical relationship. So these products of photosynthesis are going to be the reactants for cellular respiration, And then the products of cellular respiration are going to be the reactants for photosynthesis. So that is what is happening in terms of the molecules. But in terms of energy, energy cannot be recycled. So for photosynthesis, light energy from the sun is what is used to create this high energy glucose molecule. And when that high energy glucose molecule is broken down in cellular respiration, ATP is formed for the cells to use for the work that they need to perform. And then once they use that ATP, that energy is released as heat. And heat, remember, is disordered energy. So actually, energy is flowing through this system in one direction. So light from the sun is converted into the glucose and then released as ATP and then released as heat. Like photosynthesis, cellular respiration is also a type of redox reaction. And redox stands for reduction and oxidation. And remember, these two reactions deal with the transfer of electrons, which relates to the transfer of hydrogen atoms, and also relates to the transfer of energy. So in cellular respiration, the reactants are glucose, oxygen, and they get converted to carbon dioxide and water. And in this case, glucose is going to be converted to carbon dioxide. So in this process, glucose, which is a high energy, has high potential energy. Glucose is composed of carbon, hydrogen, and oxygen, and carbon dioxide only has carbons and oxygens. So what will happen to the glucose is that these hydrogens are going to be removed. And oxidation is the type of reaction which involves the loss of hydrogen atoms, the loss of electrons, and the loss of energy. So during cellular respiration, glucose is going to be oxidized into carbon dioxide. And redox reactions always occur together. So if something is being oxidized, something else is being reduced. And in this case, oxygen is going to be converted into water. And if you notice, oxygen is only composed of oxygen atoms. Water is composed of oxygen and hydrogen atoms. So in this case, oxygen is going to gain hydrogen atoms, so it's going to be reduced. And remember, reduction is the opposite of what you would think. Reduction is the gain of hydrogen atoms, the gain of electrons, the gain of energy. So oxygen is going to be reduced. Now this process of oxidizing glucose, so oxidizing is removing the energy from glucose. Remember, glucose is a high energy molecule. It has a lot of potential energy. And so the process of oxidizing it involves removing all this energy and using that energy to produce ATP. Well, the process of cellular respiration involves many steps, and that is because this energy is going to be reduced in very small increments. So that glucose is going to be broken down a little bit by a little bit, releasing that energy a little bit at a time. And that's important because that allows the cell to control the amount of energy that is released and to be able to use that energy for work in the cell. And an analogy I've thought of is your car. In your car, you have a tank of gas. If it's a full tank of gas, you have 10, 15, 20 gallons of gas in your car, and that is a lot of potential energy. And if you want the car to move, you want to release that energy a little bit at a time. So in your engine, so that gas is going to be burned. It's going to go through combustion a little bit at a time because then it's controlled release of energy, and that allows the car to do the work of moving forward. If instead you decide to release all the energy in your tank of gas at a time, that would be an uncontrolled release of energy that would probably destroy your car, and that energy would not be available to do the work of the car, which is to move. So like the cell wants to be able to have control of the energy as it's released, that potential energy as it's released from the glucose molecule. So it's going to oxidize the glucose molecule a little bit at a time so that it maintains control over that energy. The mitochondria is considered the powerhouse of the cell, and this is where a lot of the reactions involved in cellular respiration occur. And so you should have a good idea of the anatomy of the mitochondria. First of all, the mitochondria is composed of two membranes. There is an outer membrane and an inner membrane, and the outer membrane is pretty smooth. It surrounds the whole organelle. The inner membrane has all these folds. So the inner membrane has all these folds, and they are called cristae. And they increase the surface area of the inner membrane, and that is important because that's where you find the electron transport chain. And the electron transport chain is the part of cellular respiration that makes the most ATP. Also, in the structure of the mitochondria, you have the intermembrane space. That is the space right between those two membranes. And then there is the matrix, which is the liquid inside that inner membrane. So that's the liquid inside the inner membrane. And that is important because that's where the citric acid cycle takes place. The overall process of cellular respiration involves 4 phases. And those phases are glycolysis, preparatory reaction, the citric acid cycle, and the electron transport chain. And remember, the overall process of cellular respiration is to oxidize glucose. So oxidize glucose, remove all the energy from glucose through these reactions, and the end product is ATP. So as glucose is being oxidized, that energy is going to be removed, and ATP will be directly made in several of the phases, but also electron carriers are going to be generated. And those electron carriers are NADH and FADH2. The electron carriers that are involved in cellular respiration are NADH and FADH2. And these are molecules that are going to be reduced during the process of cellular respiration. So they are going to be reduced, which means they gain electrons, they gain hydrogen atoms, and they are gaining energy. So this shows the 2 molecules that will be reduced. So for the NADH, it starts out as NAD, and when a hydrogen is added to it, when it gains a hydrogen, gains an electron, gains energy, then it is converted into NADH. So NAD did not have a hydrogen. Now that NADH has a hydrogen, it is now reduced. FADH2 is similar. FAD starts out here. This is FAD, and a hydrogen will be added. Actually, 2 hydrogens will be added. And when the 2 hydrogens are added, now this is the FADH2 molecule. Okay, so now the FAD is reduced into FADH2. So again, it gained electrons, gained hydrogens, it gained energy. So these electron carriers, you should think of them as energy carriers. So during the process of cellular respiration, as the glucose is oxidized, these electron carriers will be reduced. So as energy is removed from the glucose molecule, that energy is then added to these carriers, which carry that energy to the electron transport chain. The first phase of cellular respiration is glycolysis. And if you look at the name of glycolysis, it tells you basically what is happening. Glyco refers to glucose, and lysis means to split. So in the overall reaction of glycolysis, you're going to start out with a glucose molecule, which is a 6-carbon molecule, and basically split it in half, which will produce 2 pyruvate molecules, and they're both 3-carbon molecules. So glycolysis is basically the splitting of glucose into 2 3-carbon molecules called pyruvate. And this occurs in the cytoplasm of cells, and it's a universal reaction. Most organisms use this, so it was probably the first heterotrophic reaction that evolved. Heterotrophic, remember heterotrophs have to eat others to get their organic molecules. Now, this reaction is deceptively simple because actually going from glucose to pyruvate, this is going to involve 10 different reactions. So as I mentioned before, during the process of cellular respiration, glucose is going to be oxidized in small increments. So as you go through this process, energy is released in small amounts. And during the process of glycolysis, NAD and ATP are going to be produced. The overall process of glycolysis is to break glucose in half to form 2 pyruvate molecules. And this seemingly simple process actually involves a series of 10 reactions. And there are 2 basic steps involved in these reactions. The first step is the energy investment step. And that involves actually using 2 molecules of ATP to destabilize the glucose molecule so it can break in half. When I talked about ATP before, I mentioned that ATP is the readily usable energy of the cell. And the way ATP works is ATP, adenosine triphosphate, has 3 phosphates. That last phosphate to phosphate bond is very unstable, has a lot of potential energy. So what will happen is the ATP will break off that phosphate and it will form ADP, adenosine diphosphate. And then that third phosphate is going to bind to a molecule and change its shape. So what happens during the energy investment stage is that 2 ATP molecules are going to phosphorylate the glucose molecule. So they're going to release that third phosphate. That third phosphate will bind to the glucose molecule. That will destabilize the glucose molecule so it splits in half. And the resulting molecules, these 3 carbon molecules with a phosphate on them, are actually G3P. So G3P, and it's just interesting to note that G3P is also the product of the Krebs cycle in photosynthesis. So in photosynthesis, you end up producing G3P, and G3P can then be converted into glucose and starch and other molecules. The second stage of glycolysis is the energy harvesting stage. And this occurs after the energy investment stage. So energy investment stage, this involved using 2 ATP to phosphorylate the glucose molecule, which destabilized it and caused it to break into 2 G3P molecules. Now, each of those G3P molecules is now going to be oxidized through a series of reactions. So oxidized means energy is going to be removed, hydrogens are going to be removed, and that's energy harvesting. And that energy that is removed is then going to be used to reduce NADH or to produce ATP. So for each G3P molecule, 1 molecule of NADH is going to be produced and 2 molecules of ATP. So because there are 2 G3P molecules, that means you will generate 2 NADH molecules and a total of 4 ATP molecules. So the total energy production for glycolysis is going to be a net gain of 2 ATP, because remember 2 were used during the energy investment step, and then 4 were produced during energy harvesting. So your net gain is 2 ATP. And also you will gain 2 NADH. So that's the energy harvesting stage, and at the end of that stage, the molecule is in the form of pyruvate. So there will be 2 pyruvate molecules at the end of glycolysis. Substrate-level phosphorylation explains how ATP is produced during glycolysis, and this involves the use of an enzyme. And this also involves phosphorylation, so this involves phosphate groups binding to molecules and changing their shape. So during glycolysis, one of the intermediates, the organic molecules, which is phosphorylated, it has a phosphate group bound to it, it will transfer the phosphate group to the enzyme, and then the enzyme transfers that phosphate group to ADP, which will form the ATP. So during glycolysis, when ATP is formed, that involves an enzyme, which is phosphorylating the ADP. Remember, phosphorylation means adding a phosphate. The second phase of cellular respiration is called the preparatory reaction, and this involves preparing pyruvate to enter into the citric acid cycle. This is the preparatory reaction, and this involves taking pyruvate and converting it into acetyl-CoA. So acetyl-CoA will be able to enter the citric acid cycle. And during this process, NADH is going to be produced, and also carbon dioxide as a waste product. So during this process, pyruvate is going to be oxidized into acetyl-CoA. So it's going to lose a hydrogen, lose some energy, and that will involve reducing NADH, because remember, whenever you have an oxidation reaction, you're going to have a reduction reaction. So the pyruvate will be oxidized to convert it into acetyl-CoA, and that will reduce NAD into NADH, and it will also produce carbon dioxide as a waste product. Once pyruvate has been converted by the preparatory reaction into acetyl-CoA, now it can enter the citric acid cycle. And the citric acid cycle is the third phase of cellular respiration. This is a simplified diagram of the citric acid cycle, and in this cycle, the acetyl-CoA, which is a 2-carbon molecule, is going to combine with a 4-carbon molecule called oxaloacetic acid. So this molecule is then going to form a 6-carbon molecule called citric acid. And of course, this gives the name to the cycle. And during this process, the citric acid is going to be oxidized, energy is going to be removed, and then that's going to result in reducing NAD and FAD into NADH and FADH2, 2 types of electron carriers. It'll also produce an ATP, and the waste product is going to be carbon dioxide. This is another example of the citric acid cycle, and this shows the preparatory reaction which produce the acetyl-CoA, and this will go in and combine with a 4-carbon molecule. So the 2-carbon acetyl-CoA is going to combine with the 4-carbon molecule, forming the citric acid. And so the 6-carbon molecule is then going to go through the whole cycle, and as it goes through, it is going to be oxidized, electrons are removed, hydrogens are removed, energy is removed. And as those electrons, hydrogen atoms, and energy is removed, as it is oxidized, it is going to reduce NAD. So it's going to reduce NAD a couple of times. So there's the NAD, and it will also reduce FAD into FADH2. So these are the electron carriers. Think of them as the energy carriers. So as energy is removed from the citrate, as the citrate is oxidized, that energy is transferred to these electron carriers. And also during this process, ATP will be formed, and ATP is going to be formed by that substrate-level phosphorylation. And again, that is where an enzyme is going to add a phosphate group to an ADP molecule. So this is how the energy is being removed during the citric acid cycle. Also, as the energy is being removed, those hydrogens are being removed. Carbon dioxide is released as a waste product. So carbon dioxide is going to be released as a waste product, and then as an end part of the cycle, since it's a cycle, that 4-carbon molecule needs to be regenerated. So it needs to be regenerated, and as it is regenerated, you generate another NADH molecule. And so then that 4-carbon molecule is regenerated, renewed, and now it's ready to combine with the next acetyl A that comes into the process. The electron transport chain is the fourth phase of cellular respiration, and this is a little bit different than the first three phases of cellular respiration. The first three phases involve biochemical reactions where enzymes are converting substrates into products. But the electron transport chain is actually a physical structure found on the cristae of the inner membrane. So the inner membrane has all these folds called cristae, and the electron transport chain is embedded in this membrane. So the electron transport chain is composed of proteins embedded in the inner membrane. In the inner membrane, it's called a membrane because it has membrane structure with a phospholipid bilayer. These proteins that are part of the electron transport chain, these are all redox proteins. So redox, reduction, and oxidation. And what that means is that these proteins are able to gain electrons and lose electrons. So when the electron carriers, NADH and FADH2, go into the mitochondria, they are going to release their electrons to these redox proteins, and those electrons are going to be passed from protein to protein to protein. So they're going to flow along the electron transport chain. So as the electrons are dropped off from the NADH and the FADH2, those electrons are going to flow along these proteins. So they're going to flow along the proteins. So the NADH, FADH2 are going to lose their hydrogens. They're going to be oxidized, and that will cause them to form NAD and FAD. And those NAD and FAD molecules are going to cycle back to glycolysis in the Krebs cycle to pick up more electrons. So those electrons are dropped off by the NAD and the FADH2. Those electrons are transported along the chain. And as the electrons flow along the chain, that's going to provide energy for some of these complexes to pump hydrogen ions out into the intermembrane space. So some of these redox proteins are actually called proton pumps because they're going to pump hydrogen ions, and a hydrogen ion is a hydrogen atom without an electron, which is a proton. So they're going to pump these hydrogen ions out into the intermembrane space. And those hydrogen ions come from the NADH and the FADH2 molecules. When they drop off their electron, a hydrogen ion is going to be released. Hydrogen ion is released, and then that's the source of the hydrogen ions that will be pumped out to the inner membrane space. Now the electron, when it is done flowing along the electron transport chain, it needs to go to a final electron acceptor. So it needs to go to its final resting place. And that final resting place is, it will combine with hydrogen ions and water and oxygen to form a molecule of water. So this is where oxygen is required in the process of cellular respiration. The process by which the electron transport chain produces ATP is called chemiosmosis. And this is the same process that is involved in photosynthesis. And this is a dynamic process that involves the electron transport chain. So remember, the electron transport chain is a series of redox proteins that are embedded on the membrane of the cristae in the mitochondria. So for orientation, this would be the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. This is the intermembrane space, so the space between the two membranes, and this is the matrix. This is the liquid that you find inside the cristae or interior to that inner membrane. So these redox proteins are embedded on the cristae, and the way the electron transport chain functions is that the electron carriers, NADH and FADH2, that were generated during glycolysis, the preparatory reaction, and the citric acid cycle, these electron carriers will carry their electron to the redox proteins, and they will transfer their electron to the redox proteins, and in doing so, they're going to lose the hydrogen ion. So they're going to lose their hydrogen ion, and NAD will be formed, so it's losing the hydrogen atom, so it's going to be oxidized. So the NADH and the FADH2 are going to be oxidized. They're going to release hydrogen ions here. The electrons are going to the electron transport chain, and then these electron carriers are going to go back to glycolysis, preparatory reaction, and Krebs cycle to pick up more electrons, more hydrogens, more energy, and bring it back to the electron transport chain. So that is how the electron carriers are involved. The electron, once it is dropped off to those redox proteins, it's going to be transferred from protein to protein to protein through those redox proteins, and when it is completed, when it has finished going through the electron transport chain, it's going to combine with oxygen to form water. That flow of electrons through those redox proteins is going to power some of the complexes to pump hydrogen ions out to the intermembrane space. So some of these proteins are called proton pumps because a hydrogen ion is just a proton. And these proton pumps are going to pump the hydrogen ions across the membrane. And this is a form of active transport because you are moving molecules from an area of low concentration to an area of high concentration. So that will result in building up a very high concentration of hydrogen ions in this intermembrane space. Now, to produce ATP, that involves the ATP synthase. So this is the ATP synthase, and the ATP synthase is going to allow passive transport of these hydrogen ions through the membrane. So part of the ATP synthase acts as a channel, and it will allow passive transport of the hydrogen ions back through the membrane. So this high concentration of hydrogen ions is going to flow through the ATP synthase, and that will allow ATP to be generated. So ATP, the phosphate, will be added to the ADP, and that will produce ATP. So this is the process of chemiosmosis. And during cellular respiration, this is where you have the highest amount of ATP produced. Cellular respiration requires oxygen, and it's important to know the actual role of oxygen in this process. And oxygen is only involved during the electron transport chain. That's the only time that oxygen is involved. And in the electron transport chain, remember that is a complex or a series of protein complexes embedded in the membrane, the inner membrane of the mitochondria. And the electrons are going to be passed from molecule to molecule, and when the electron finishes being transported through the electron transport chain, it needs to go to a final electron acceptor. So I think of that as its final resting place. So once the electron, after it has been dropped off by NADH or FADH2, it's going to be transmitted through the electron transport chain, and then it has to go to its final electron acceptor. And in aerobic cellular respiration, that is oxygen. So the electron is going to combine with oxygen and two hydrogen ions to form water. So oxygen is actually very important because that is where the electron is going to go at the end of the electron transport chain. And if oxygen is not present, then the electron transport chain and actually the Krebs cycle will not function. This figure summarizes the ATP production in cellular respiration, and it shows how all the different phases relate to producing ATP. The first phase of cellular respiration is glycolysis, and glycolysis occurs in the cytoplasm, and the purpose of glycolysis is to take glucose and to produce 2 pyruvate molecules. And during this process, there's going to be a net gain of 2 ATP, because remember there is an energy investment phase that utilized 2 ATP. So your net gain is 2 ATP, and also there is the production of NADH. After glycolysis, there's the preparatory reaction. Preparatory reaction converts the pyruvate molecules into acetyl-CoA. So you generate 2 acetyl-CoA molecules, and they are able to enter into the mitochondria. And during this process, 2 molecules of NADH will be produced, 1 for each of the acetyl-CoA. The acetyl-CoA are then able to enter into the citric acid cycle, which is the 3rd phase of cellular respiration. And those organic molecules are going to go through the cycle and be completely oxidized. So all the energy is going to be removed. And there will be 2 turns of the citric acid cycle, 1 for each of the acetyl-CoA molecules. So in total, there will be 6 molecules of NADH produced and 2 molecules of FADH2. And in addition, you will have 2 molecules of ATP produced. All of the ATP produced so far is produced by substrate-level phosphorylation. So that involves an enzyme adding a phosphate group to an ADP molecule. Now, all of these electron carriers that are generated during the first 3 phases are going to go to the electron transport chain. So they are transporting their electrons, transporting their hydrogens, transporting the energy to the electron transport chain. When those electrons are dropped off, then they will be transported through those redox proteins embedded in the membrane. Redox proteins are going to activate proton pumps, which perform active transport to build up a high concentration of hydrogen ions on one side of the membrane. Then the ATP synthase is going to allow those hydrogen ions to pass through using passive transport into the membrane, and that motion of the hydrogen ions flowing through the ATP synthase is going to generate ATP. And that process is chemiosmosis. So these ATP are produced by chemiosmosis, whereas these ATP are produced by substrate-level phosphorylation. If you add up all of the potential ATP that could be produced, ideally, the ideal amount of ATP is 38 ATP that could be produced per 1 glucose molecule. Fermentation is a type of carbohydrate metabolism that occurs when oxygen is not present. And when oxygen is not present, we refer to that environment as anaerobic. Anaerobic. An means without, aero means oxygen. So in anaerobic environments where oxygen is not present, cellular respiration cannot take place. Remember, oxygen acts as the final electron acceptor at the end of the electron transport chain, and if the electron transport chain can't function, cellular respiration can't function. So as an alternative, if oxygen is not present, some cells can perform fermentation to generate ATP from glucose molecules. And fermentation is basically a modified form of glycolysis. So in glycolysis, remember you start with a glucose molecule, 6-carbon glucose molecule. It runs through a series of reactions and the end product is pyruvate. In fermentation, you also start with a glucose molecule. It runs through a series of reactions and produces pyruvate. And then at the end of fermentation, that pyruvate can be converted into another end product. So it could be lactate is one type of fermentation, lactic acid fermentation, or alcohol fermentation. So those are 2 types of fermentation. Lactate fermentation, lactic acid fermentation can be accomplished in animals and bacteria. Alcohol fermentation is usually in plants and yeast. And one of the differences between lactic acid fermentation and alcohol fermentation is with alcohol fermentation, you get the release of carbon dioxide. In lactic acid fermentation, there is no carbon dioxide produced. But fermentation has an ideal output of 2 ATP. So remember, this is like glycolysis. So there's actually an energy input stage and an energy generation stage. And so your net gain of ATP per 1 glucose molecule is only 2 ATP when performing fermentation. Cellular respiration, the ideal output is 38. Often it's less than 38, but the maximum you can get is 38. So fermentation has a lot fewer ATP produced than in cellular respiration. With fermentation, there are lots of different types of fermentation. I mentioned alcohol fermentation and lactic acid fermentation, but there are many different types of fermentation. And fermentation is pretty important for humans because it produces different products that we are interested in. Of course, alcohol fermentation produces alcoholic drinks like wine and beer. Lactic acid fermentation, produced by various types of bacteria, produces yogurt and cheese. Acetic acid fermentation is what is used to produce vinegar. And alkaline fermentation can produce some types of food like Japanese natto, which is fermented soybeans. Animal cells have the ability to perform cellular respiration or fermentation, and this is especially important in muscle cells. So all of our cells are able to perform cellular respiration or fermentation, but this usually comes up in muscle cells, especially when muscle cells are being used to a great extent. So let's say you have a person who is playing a game of basketball or running a marathon. So normally the muscle cells are going to need a high output of ATP. So the most efficient breakdown of glucose is to perform cellular respiration, where the glucose goes through glycolysis. Pyruvate is generated. Pyruvate goes through the preparatory reaction, enters the mitochondria, goes through the citric acid cycle, and then it goes through the electron transport chain and produces a lot of ATP. And so for the average human cell, out of 1 glucose molecule, you will generate 34 ATP, which is a pretty good yield. But as the person's muscles are being used, a lot of ATP is required, and often the oxygen supply is not sufficient to support the electron transport chain. So in that case, the muscle cell is going to switch its metabolism from cellular respiration to fermentation. So now the pyruvate is going to be converted into an end product of fermentation, and in this case, it would be the lactic acid. And when this happens, this is a process of fermentation, now the cell is only going to produce 2 ATP from 1 glucose molecule. And while the yield is very low, at least there is still production of ATP with no oxygen supply. Because without the oxygen, none of the cellular respiration is going to occur. So you're not going to get the electron transport chain occurring. So when muscles are being used a lot, when there is a high demand for ATP and the oxygen supply cannot support that high demand, the cells switch over to fermentation, producing ATP and lactic acid. And the evidence that your muscle cells have generated lactic acid is that they become sore afterwards. Metabolism is defined as the sum of all the biochemical reactions that occur in an individual. And those reactions can be either catabolic reactions or anabolic reactions. Catabolic reactions involve breaking down large molecules into smaller ones, and that usually involves an output of energy. Anabolic reactions involve taking smaller subunits, building larger molecules from them, and that usually requires a net input of energy. Cellular respiration is one of the major catabolic reactions that occurs in the cell. So cellular respiration involves glucose being broken down to produce ATP. And that is the usable energy for the cell. This is going to do the work of the cell. And in terms of metabolism, your metabolism is able to adapt to the types of macromolecules that you have. So for instance, role of carbohydrates in our diet is to provide glucose so that our cells can perform cellular respiration and produce ATP. But if for some reason you don't take in enough carbohydrates, then the fat in your diet or the fat that has been stored in your body can also enter this process. The fat can be converted to pyruvate, and then cellular respiration can take place. Also, if, for example, you don't have any carbohydrates or fats available, proteins can also be broken down, and they can enter the cellular respiration pathway at a couple different points, and in this way provide ATP for your cells. So cells are able to adjust to the environment, adjust to the available molecules to produce ATP. Then also the interesting thing about metabolism is a lot of these reactions are reversible. So for example, if carbohydrates are needed, this process, pyruvate, can be taken and it can be used to produce carbohydrates. Or if fats are required or you're ingesting too many carbohydrates, you don't need all that energy, the pyruvate can be used to produce fats. And also if you need proteins produced, some of the different components involved in cellular respiration, like acetyl-CoA, can be used to generate amino acids. And amino acids are really important for the formation of proteins, and many of those proteins act as enzymes. And if you remember when I talked about amino acids, there are 20 amino acids that living things use to produce their proteins, and 11 of those can be made in our own biochemical processes. And so those are called non-essential amino acids because our own cells can make them. But there are 9 amino acids which we call essential because we are unable to make them in our cells. So we have to take them in through our diets. And often a deficiency of an essential amino acid can lead to a disorder. In a previous lecture, I discussed the process of photosynthesis, and this lecture went over the process of cellular respiration. And it's really important for you to see the similarities in the processes and where they complement each other. So first is in the organelles. You should know the structure of the chloroplast and the mitochondria and how they are similar, that they have double membranes. They both have an electron transport chain that can perform chemiosmosis. So there's a similarity there. Then you should also know the flow of energy. So solar energy flows through the light reactions or goes to the light reactions, which provides energy for the Calvin cycle, which produces the carbohydrates. And then in cellular respiration, those carbohydrates are then used to go through glycolysis, preparatory reaction, citric acid cycle, electron transport chain, and the whole purpose is to generate ATP for the cells. And just the interesting thing in terms of the organic molecules, so in the chloroplast, CO2 is converted into an organic molecule, a carbohydrate, and that process, CO2 is going to have a hydrogen added to it, so it's going to be reduced. So CO2 is reduced into a carbohydrate, and that is going to oxidize NADPH. Whereas in the mitochondria, that carbohydrate is going to lose its hydrogen. It's going to be oxidized into carbon dioxide. And in that process, NAD is going to be reduced into NADH. This is the end of the lecture on cellular respiration. The overall process of cellular respiration is to take the high potential energy molecule of glucose and run it through a series of reactions to generate ATP, which is the readily usable energy for the cell. ATP can do the work for the cell. And in this process, oxygen is required, and the waste products that will be produced are carbon dioxide and water. And it's also important to realize how cellular respiration fits in with photosynthesis to facilitate the flow of energy through a cell or through an ecosystem.